Hello, good afternoon. We will continue our lesson on biotechnology. And so the next part is something called DNA fingerprinting. You probably have heard of this before. It's to analyze an individual's DNA, which is very unique, of course, and you can compare it to a sample to determine whether it is the same person or you can determine if the individuals are related, right? So there are various information that can come out from DNA fingerprinting. Now, the actual process is, a little bit complicated, but we will try to just show the basic principle behind it, and hopefully that will help you understand how it works. So there is this term called polymorphism. Polymorphism. That is essentially any difference in a DNA sequence that can be detected between individuals. A lot of these polymorphisms exist because of things like silent mutation. Okay, we learned about silent mutation before, right? And it could happen anywhere in the coding regions or non-coding regions, and they're just kind of scattered throughout the genome of a person. And the closer you're related to someone, the more similarity the DNA is going to be. So individuals that are not related, there would be more polymorphism. Having said that, though, between any individuals, we are more than 99% identical on all our DNA. These polymorphism only accounts for less than 1% of difference that is observed between any two human individuals. Now, like I said, some polymorphism can be as a result of single nucleotide mutation, right? Those would be called SNPs, single nucleotide polymorphism. Okay, but there are other kinds of polymorphism that could exist. Okay? They are called short tandem repeats, or STR. These are usually going to be found in non-coding regions. So an example of a short tandem repeat, short means like, a few nucleotides, and tandem means one after the other. So here we have CAG, 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 CAG. So to represent this, we will put CAG and then 4. So it tells you that the sequence CAG is repeated 4 times. Okay, so that's a short tandem repeat. So in your DNA, you might have a CAG that's repeated 4 times. In my DNA, I may have a CAG that's repeated 10 times. Okay, so there is a difference, right? So that would be an example of polymorphism. Okay, so polymorphism can exist in various forms. It can exist on an individual nucleotide basis. That would be a SNP, single nucleotide polymorphism. It could exist because of different lengths in STR. Okay. Now, sometimes these STR can actually contribute to disease. So Huntington is such example. There is what we call like the HTT mutation, which causes Huntington disease. And in this mutation, it involves a short tandem repeat that is consists of C, A, and G. So normally on this gene, the CAG is repeated anywhere between 10 times and 35 times. Okay. And in people with Huntington disease, the CAG segment is repeated more than 35 times. So that tends to be the cutoff. If you have more than 35 repeats, right, anywhere from 36 to more than 120, then the person is going to develop Huntington disease. And for those of you who don't know what Huntington is, it's a neurodegenerative disorder that causes you to slowly lose motor functions. Okay. Now, not all STRs, short tandem repeats, are linked to disease. And there are many STR that are scattered throughout the genomes. For example, person 1 might have AGAT repeated 12 times. Okay, so if it's repeated 12 times, how long is this segment in total? AGAT repeated 12 times. How long is the whole thing? Okay, that would be 4 times 12, which is 48, right? If it's GATA repeated 15 times, that's going to be 60 nucleotides. And then if it's like, CCAC repeated 30 times, that would be 120. For someone who have the same STR but repeated different number of times, right? So just 5 times here, that will only be 20 nucleotides long. Same in this particular position, so 60. And this one is going to be 80 nucleotides long. Okay, so you can see even though they have the same STR, the same repeating units, sometimes it's longer than others, right? Depending on the person. So these STR are unique to every person. That's why we call it DNA fingerprinting. Okay, so like for person number 1, the numbers are like 48, 60, 120. But for another person, it's 20, 60, 80. So if you look at a lot of STR, each STR generates a unique number that will be unique to a particular person when it's considered as a whole set. So how do we know? Like, how do we determine the length of the DNA, right? Well, we use something called gel electrophoresis. Gel electrophoresis. Okay, so it basically goes like this. You are going to create a gel. Right? So this gel is called an agarose gel. Agarose gel. It's kind of like a milk powder, comes in a bottle. And then you would add it to a solution and you microwave it, and then it becomes this jello-y looking thing. And then you put it in a cask and wait for it to dry. Okay. So the more powder you add, the more dense the gel is going to be. And the denser the gel, the better it is at separating DNA based on their length. Okay, so sometimes it's hard to distinguish DNA that are like 20 units long versus 25. For something like that, you would need a gel that is really, really dense. Okay, so basically how it works is you would put the gel in a solution, a buffer solution, and you would apply electric current through the solution. Okay. We would start at the negative terminal and then apply current towards the positive terminal. And then on the gel, you're gonna have some wells to put your DNA sample in. Okay, so you lower your sample in here and you run the current. Okay, so the DNA is gonna get pushed down each of the lanes. Okay. Now the agarose gel is like a mishmash, a network of sieve, if you would. Okay, so the more powder you use, the finer the sieve is going to be. So the DNA is going to get pushed through these networks. If it's a really, really long DNA, then it's going to get tangled up easily, and it's not going to move down the gel too far, right? So for longer DNA, they tend to be closer to the starting point. But if it's a really short DNA, they would have no problem navigating through the network, and they tend to end up closer to the bottom. So this is what the actual experiment looked like. The gel is here, and you use a pipette to load the DNA. You have to add a little bit of dye to the DNA to help it sink into the well. That's like a blue dye usually. And then you would run the current in this direction from negative to positive, because the backbone of the DNA is negatively charged, right? Because of the phosphate, right? Sugar phosphate backbone. So the negative current will repel the DNA and push them down the lane. And so this is one lane, right? So the DNA sample here will travel down this lane, and then you have another DNA sample that's going to go down the lane. And depending on whether it's long DNA or short DNA, right? Longer DNA will stay, closer to the starting point. and then shorter ones will be able to go down further. Now you can't visualize the DNA, you cannot see them as they are going down the gel, okay? So that's what the dye is for. The dye will migrate down towards the positive end along with the DNA, and it gives you kind of like a rough idea where the DNA is going to be. Okay, so if you see the blue dye here, you know your DNA it's somewhere around here. Okay, if you keep on running the current, then this blue dye will eventually exit from the other end, and that will be too long. Okay? That means your DNA also exit, and then you won't be able to see anything. So let's say you stop the gel here, you stop the current, then you actually have to stain the gel with something called ethidium bromide. So ethidium bromide is a molecule that would insert itself into the DNA, and when you shine UV light at it, it lights up. Okay, so after you expose the gel to ethidium bromide and then you shine UV light at it, then you can see, oh, there's the DNA, there's the DNA, right? And these are individual lanes, right? Lane 2 here, the sample goes all the way there. Lane 3 goes all the way here, and then lane 4, 5 over here, so on and so forth. The sample is probably further down in 8, and you can buy what's called like a ladder, okay, ladder or markers from biotech companies, and these are predetermined DNA fragments. Okay, so it will give you like a reference sheet when you buy the ladder. It will tell you, oh, this band is 1,000 base pair long, and then down here this one is 260A, and this one is 170. So then you can compare your actual DNA sample with the ladder to have an idea of how long it is going to be. Okay, so the point is this: the further down the DNA runs in the gel, the shorter it's going to be. Remember, the gel is like a network, right? Like a sieve. So if the DNA is really short, it's going to be able to maneuver through these sieve very easily and go quite far. But if it's really big, then they are not going to go very far. All right. So let's take a look at this question in your course pack. Okay. This is the first question, and we have a gel, right, four lanes here, and you load some DNA sample. It's possible that the DNA sample has multiple DNA fragments in it, right? So here is asking where, indicate the following on the gel, where the positive electrodes was located. So if this is a gel, is the top positive or is the top negative based on what I told you? Do you remember?
The top is negative.
That's right, because we apply the current from the negative terminal to the positive terminal, right? That's going to be the direction of the current, which will help repel the DNA. It pushes the DNA further and further down, right? If you reverse the current, then you will push the DNA the other way, which is bad. You will lose all your sample. It will all come out from the top, and you won't see anything. Okay. The location of the largest band. So tell me, where is the largest band located? Is it in lane A, B, C, or D? Let's do a poll here. Okay. The largest band. Which lane contains the largest DNA band? Anyone else want to try? I have 5 responses. We have 9 people here. Okay, well, the largest band is going to be right here in lane A. That's the largest band. Longest DNA. remember, if it's really long, it's going to have a hard time going down the lane, all right? Because it cannot navigate through those tiny holes, right? Okay. The smallest band will be right here. Shortest one, we'll say, shortest DNA. If it's really small, it's going to be able to get really, really far. Okay? If you don't understand, you should ask, because the rest of the lecture is going to be based on this concept.
I'm sorry, I joined late, so I'm a bit confused.
Okay, so we are trying to separate DNA based on their size. That's the purpose of gel electrophoresis. Okay, this is gel electrophoresis. Okay, and the purpose is to separate DNA based on size. Okay, so the gel is made up of a network like this. Okay, so if it's a really short DNA, I was saying they could go through these tiny holes, no problem, and they could go really, really far. That's why the further down it is, the shorter the DNA. If it's really, really big, then it's not going to get very far. That's why the largest band is going to be closer to the start, to the top. Okay, that's basically the gist of it. All right, let's take a look at E here. The number of cuts that were made on the plasmid to produce this number of bands. So let's do that first. Okay, number of cuts made to the plasmid. Okay, so a plasmid is a circular DNA, right? We learned about plasmid last class. We see 4 bands here. 4 bands here. So how many times do you have to cut a plasmid to create 4 bands? Think about it. If you take a circle, how many times do you have to cut it to create 4 bands?
3 times.
Okay, so I hear 2 times, I hear 3 times. Anyone else?
2 times.
2 times.
Yeah.
Well, let's see. If I cut it once and I cut it twice, right? That's gonna give me 1 band, 2 bands, right? We're gonna have 2 fragments. 2 fragments correspond to 2 bands, right? So 2 cuts is not enough to get 4 bands. So let's try again. How many cuts do we need to create 4 bands?
That'll be 4.
Okay, let's see. 1 cut, 2 cuts, 3, 4 cuts. So that's 1 fragment, 2 fragments, 3 fragments, 4 fragments, right? So 4 cuts is gonna give me 4 fragments. Okay, so if these are all plasmid, right, then it will be like 4 cuts, 3 cuts, 5 cuts, and then 4 cuts again. So this is for plasmid. All right. If it's linear DNA, right, linear DNA, how many cuts do I need to have 4 bands?
3 cuts.
That's right. Chop, chop, chop. That's 1 cut, one fragment, 2 fragments, 3 fragments, and 4 fragments. So if it's linear DNA, you need 1 less cut to achieve the same result. Does that make sense? Again, you need to ask questions if you don't know what's going on. It's only going to get more confusing as we progress. So that's gel electrophoresis. Okay, we can use it to separate DNA based on size. Now, before this, we were talking about STR, right? And we're talking about short tandem repeats. So let's take a look at how we can use STR to identify individuals. Okay, so we're gonna say STR analysis. Okay, you should copy this down. So we have individual A here. Okay, we'll call him, what should we call him? We'll call him Bob. Okay, so let's take a look at 3 chromosomes from Bob. Okay. Chromosome 2, chromosome 4. Chromosome 2, whatever, 6, and maybe 18. Okay, so these are chromosome numbers. And because we have the Human Genome Project done, completed, we would have no problem identifying where the STRs are, right? Okay, so let's say over here, there is an STR, a short tandem repeat for Bob right here. And that is going to be, CCG, totally making this up. And it's repeated 10 times. He might have another short tandem repeats here. And this one is like AT repeated 3 times. And then there is another one maybe on chromosome 18. And this one is GAA repeated, 5 times maybe. Okay. So let's say we have another person. And we have another person here. Okay. This is Alicia. Okay. And we will be looking at the same chromosomes on Alicia. Okay, chromosome 2, chromosome 6, and chromosome 18. Okay. So the same tandem repeats would be found in the same locus, same location. So CCG. Now, because they are not related, their tandem repeats are not going to be the same. Okay. So maybe for Alicia, this is repeated, 15 times. You know what? Let's repeat like, give me a second. 4 times. Okay. And then over here, we're going to have AT, and this one repeats, 10 times maybe. Okay. And finally, we have another one here, GAA, and this one could be repeated 5 times. All right. Now, nobody was able to answer me this morning when I asked this question. Let's see if you guys are more responsive now. If this is CCG repeated 10 times, how long is this STR?
Can I try?
Of course, please.
Yeah, 4 power 10.
Good try, not quite. So CCG repeated 10 times means CCG, 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 CCG repeated 10 times. So how many in total? Anyone? Is 30, right? 10 times 3, right? Do you guys understand this? It's important you understand this before we go further. If you don't understand, that's okay. You ask the question so I can clarify for you. Someone else probably has the same question.
No, I don't understand.
Okay, let's write it out. CCG10. It means it is CCG. That's 1 time. CCG 2 times. CCG 3 times. 4, 5, 6, 7, 8, 9, 10. That's what this means. It's tandem. Tandem means one after another. So it's CCG lining up one after the other 10 times. So how long is the whole DNA? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30. Right? Or simply, 3 times 10. That's how we got the 30. Anybody have any question? Okay, what about here? What about AT bracket 3? How long is this going to be?
6.
6, that's right. 3 times 2, right? That's going to be 6. Okay, this one is going to be 3 times 5, which is 15. Okay, so similarly over here, we have 4 times 3, which gives you 12. 10 times 2 gives you 20. And then 5 times 3 gives you 15 again, right? So let's say we use PCR to clone these STR. Okay, we know their location. We can design primers that are next to the STRs, and we can run PCR to amplify them. So after we amplify them, we can load them on a gel. Okay, so this is a gel. Okay, and these are the lane. Yeah, so we're gonna take Bob's sample and we're gonna lower it in lane number 1. We're gonna take Alicia's sample and we're gonna lower it at lane number 3 here. We're gonna hook it up to the negative terminal here and the positive terminal here to push the DNA down. Okay, so let's take a look at Bob's sample. Okay, now how many bands are we going to see in Bob's sample? If we use PCR to amplify 3 STRs, then I will see 3 bands. Okay, this band is 30 units long, 30 nucleotides long. Do you expect it to be at the top, in the middle, or at the bottom? Look at the 3 bands we have. Top, middle, or bottom?
Top.
Top, that's right, because it's the longest one, right? So it's going to
It's
have a hard
largest.
time going through. Yeah, it's the largest, exactly. It's going to have a hard time going through the mishmash, right? So you're going to expect to have a band at the top here. Okay. Now in reality, right? Like the bands are all like no colors, right? I showed you that in the PowerPoint, huh? Like they just gonna give a white band if you shine UV light at them after staining them with ethidium bromide. But I'm just color coding it to help you understand. Okay, what about the red one? Well, the red one is 6. It's really short, so it's gonna go really, really far, and it's probably gonna be somewhere down here, right? And 15 is probably somewhere in between, not necessarily like halfway. Right? It's not like a linear relationship, right? But certainly is going to go further than the 30, but not as far as the 6. Do you guys understand this? Now, if you're copying this, I'm going to give you a moment to try to come up with the bands for Alicia. Okay, and then I'm gonna talk about it with you afterwards. Okay, I'm gonna give you like a minute or 2 to try that out. Okay. This one is 12. So 12 is going to be you know, quite low. And don't forget, we have to look at these one as well relative to these. So if this is where the 15 is, 12 is going to be like very similar. Okay, maybe slightly lower. Okay, but it's going to be at a very similar position, right? Alicia also has a 15. So that's going to be in the exact same location as Bob's. And then the red one is 20. So it's going to be further up. okay? But not as high as the 30. Do you understand? So Bob's, just looking at 3 spots, his band patterns are going to be very different from Alicia's, right? So you can tell them apart. If I find a sample, I don't know, at a crime scene or something, and then I see 3 bands like this, then we're like, oh, it's Bob's sample, not Alicia's. Okay? But in real life, the real DNA fingerprinting, we are not just gonna look at like 3 bands. We're gonna look at all the STR that exist, and you're gonna get a picture like this, right? So all these bands, right? That's basically, each band corresponds to a particular STR that exists in the human body, right? So you can see this guy right here, this lane, that looks exactly like this band, like this lane here, right? So these 2 samples are probably from the same individual. You know what I mean? Okay, so I have another band here, another lane. That looks very similar to this one. So that is probably from the same individual as well. Okay? So essentially, that's how DNA fingerprinting works. Right? When you have many STRs, you're looking at it, then each person is going to have a unique banding pattern, right? Like a unique barcode made up of various STRs. Okay, and that is unique to that person. Any question? Okay. Let's go back to here. The plasmid is treated with ECOR-R1 and BAM-H. When the digested plasmid is separated using gel electrophoresis, how many bands will you see? Okay. Let's take a look here. Let me launch the poll for you. Okay, thank you. So if you treat it with ECOR-R1, that means we are going to cut right here, 1 cut, 2 cuts. So that's 1 band, that's 2 bands. You have 2 fragments. Right? This one is going to travel further down the gel this one is going to travel further down the gel because it's shorter, right? Compared to this one. This is the longer one. Let's try another one. All right. We have sample A, which contains CAG repeated 10 times. So that's a total of 30 nucleotides. This one is CAG repeated 15 times. That's going to be 45 nucleotides long. Which sample will be further down the gel when separated using gel electrophoresis? The shorter one will, right? So sample A is going to be further down because it's shorter. Okay.
Can you explain that? I'm a little confused.
Sure. First of all, Sarah, do you understand why this is 30 and why this is 45?
Yeah.
Okay. So on a gel, right? On a gel, if you load something with 30 and 45, let's say we load sample A here and sample B here, right? The shorter one will always be further down compared to the longer one, right? Think of the gel as an obstacle course, right? With like all these nets in it, okay? All these nets in it, like an obstacle course. Rope, what do you call these? Like rope ladders or whatever, right? Okay, so like think of an adult, right? And then a little kid, right? The adult will have harder time maneuvering through these holes to get to the other end, right? Okay, whereas the kid, it's much smaller, they're going to be able to go much faster, right? So if given limited amount of time, the kid will be able to get further compared to the adults. That's the same thing with the DNA. If the DNA is longer, it's not going to be able to go as far down the lane compared to the shorter DNA. Right? So the question asks which sample will be further down, which sample is going to be able to go, further. It's sample A, because the shorter it is, the farther it's going to be able to go on the gel. Does that help a little bit, or any follow-up question?
No, that helps. Thank you.
Okay, great. Anyone else? All right. Now, before we do the LFLP analysis, let's go back to the PowerPoint here and see if there's anything else we could do. Okay, this one is good. Number 10. Predict what will happen in each of the following situation during gel electrophoresis. Okay. What if the student made a mistake? Right? Instead of the black to the black and the red to the red, like when you hook up the wires, if they mixed it up, right? If they reverse the electrode so that the current runs from positive to negative, what's going to happen in that case? Okay, so that's negative, that's positive. Usually it goes this way, right? But the guy or the girl hooks it up the wrong way, then what's happening to your DNA? Well, the DNA will be pushed this way, right? So the problem is all samples will exit the gel at the top. Okay, everything is lost. Everything will be lost. It's gone. The sample is gone. You lost your DNA. And if this is the only DNA sample from the crime scene and you hooked it up the wrong way, then we have a problem. Okay, so that's the normal way. B, run the gel for too long, dye front is no longer visible. I remember the picture I showed you earlier, right? We have to add the dye to help the DNA sink into the well. I guess it's hard for you to visualize, right? Like without actually, doing it yourself. But let me try to show you again in the PowerPoint here, right? Okay, like I don't know if you probably cannot see, but there is a little well here. Okay, like when you make these gel, it literally is like it's like making jello. Okay, so like you have a little container. Okay, so you take those powders, the agarose powder, you mix it in the buffer, you put it in the microwave to dissolve it, and then you would pour that stuff into this mold here, and then you would put a comb over here. You put a comb in it, like a literal comb that has these teeth in it, and it would just like sit in the gel, right? So when the gel solidifies, when it turns from liquid to solid, much like jello again, and then you remove the comb, right? You move the comb, then the comb would leave behind little slots, right? Okay. So you would take your pipette and you pick up your DNA sample, which is mixed with a dye, and you would inject the sample right into the little hole here. So that the sample A goes here, sample B goes here, so on and so forth, right? So that when you turn it on, when you turn on the current, the DNA will run, as in like it will move down the lane, but so will the dye, right? So the dye is going to move down, and you can visually track where the dye is, right? And again, the dye gives you an idea of how long you have been running your gel. So the question is asking, what happens if the dye is no longer there, the dye front? Okay, like the dye front went all the way to the end and then it exits into the buffer, so you don't see it anymore. Like what does that mean? All right, so I told you earlier, right? If the dye front is gone, there's a good chance your DNA is also gone. Okay, remember, the DNA will move down. And sure, I said the longer one will be closer to the top, but if you run it for too long, even the long ones will exit at the bottom, right? Think about the obstacle course and the adder, right? When given sufficient amount of time, the adder will also make it through the obstacle course, right? Okay, so everything will exit at the end. If the gel runs for too long. Runs for too long means like you keep the current on for too long. Okay, usually you do it for like 45 minutes, 60 minutes, right? But if you run it for too long, then sample is gone. Okay, so if dye front is gone, sample, oops, that's too thick. Sample, exit at the bottom. Okay, everything will be lost again. Okay, everything will be lost. All right. Now, remember, on the gel, you cannot see the DNA, right? Like I show you this picture again. This is the gel, right? I post a video of like an actual gel electrophoresis experiment. You can take a look at the video resource and see how it's done. But over here, right? Oh, you can see the well quite nice here, right? Look there, there's the well, there's the well. That's where you were loading the sample, right? But like, where are the DNA? Like, I mean, we see the dye front, so we have an idea that, it's somewhere here, but we cannot see the individual DNA, right? Not until we stain it with ethidium bromide, this stuff will stick itself into the sugar phosphate backbone. And when you shine UV light at it, you actually detect the gel and you put it in this tiny black box that exposed UV to it, you will be able to see the DNA light up, okay? So if you forget to add the ethidium bromide, what's going to happen? What's going to happen is you cannot see the DNA at all. Cannot see the DNA. Even with UV, right? You won't be able to see it. Now, this stuff is toxic, okay? It inserts itself into the DNA, right? So you don't want to be like touching it without gloves, because like if it gets into your body, it's going to bind to your DNA and it's going to cause mutation, right? So that's not a good thing. Anyway, that's gel electrophoresis. I hope that makes sense. Okay, the next thing we're gonna talk about is RFLP. And I'll try to explain it on another diagram here. Okay. RFLP. Okay, so RFLP is another kind of polymorphism. In case you forgot what a polymorphism is, it's basically detectable DNA differences between individuals. You can have polymorphism on a single nucleotide level. That's called SNP, single nucleotide polymorphism. We can have polymorphism in short tandem repeats. The CAG, 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 repeated 10 times versus 20 times. That's a polymorphism. Another polymorphism is called RFLP, which stands for restriction fragments length polymorphism. Okay, so restriction fragment are basically fragments that you get when you treat the DNA with a restriction enzyme. Okay, and restriction enzymes recognize certain sequences and cut them, right? Okay. So let's take a look here. Suppose we have a restriction enzyme. So consider a restriction enzyme. Consider a restriction enzyme that recognize you know, AAT, ATT. Okay, so this is a palindrome, right? Okay, and suppose it's gonna cut like this. Okay, it doesn't really matter in this case whether they create blunt ends or sticky ends. Okay, so let's just say we have DNA fragments. So we have a strand of DNA. Okay, it could be the whole chromosome. It doesn't have to be. It doesn't really matter. So there it is. That's one strand of a DNA. That's the other strand of a DNA, the double helix, right? Okay. So let's say this person has AAT, ATT. Sorry, that's not a good diagram. Let's try that again. Okay, let's draw it longer here. So that is individual number 1. Okay, we will call him, I don't know, Patty. Okay, so we have AAT, ATT here. Okay, and let's say Patty has 2 of such recognition site. Okay. So what happens if you take her DNA and you treat it with the restriction enzyme? Okay. How many cuts are we gonna have for her DNA? Well, there are 2 such recognition sites, right? So cut it once, cut it twice. Okay. So her sample, if you run it on a gel, right, you're gonna get 3 bands. You're gonna have 2 short ones and 1 long one. That's Patty's sample. Now let's say we have another person. Okay. We have Joey here. Joey, this is the same chromosome. Okay. Joey also has AAT, ATT here, but he has a silent mutation that causes the T to become a C at this location. Right. So when you treat it with, sorry, this is treated with RE. When you treat it with the restriction enzyme, and then you load it, this time there's only 1 recognition site. It's going to cut it here, but not here. So he will have a super long band and then a short one at the bottom. Right? So this is another way to analyze DNA samples. Okay. You don't have to clone the DNA in this case. Okay? You can just expose the sample to restriction enzymes and then load the final product to distinguish them. Okay. Okay, we'll do one more exercise on gel electrophoresis before we move on. Okay, so there is this question right here. The recognition site for BMH and BCL1 is shown below. Okay, so BMH is going to recognize GGA, TCC. BCL1 is going to recognize this particular site. So here it says, if the following DNA was cut with BAMH, how many DNA fragments would you expect? Okay, so that's BAMH right here. And it's going to recognize GGA TCC. So let's look for GGA TCC. I'm going to give you a moment to try to identify that yourself.
I think it will be 2 fragments.
2 cuts or 2 fragments? 2 fragments, you said? Sorry.
One cut,
1
2
cut, 2 fragments. Okay, let's see. So GGA TTC, right? So there it is. GGA TCC, right? Is there an other GGA TCC? I don't think so, right? Okay, so if you treat it with BAMH, it's going to cut it right here, right? So that's going to be 2 fragments. Okay, 1 cut, 2 fragments. Same DNA. So if you treat the DNA fragments produced in part A, so like, this piece and this piece, and you're going to treat it with BCL1, what results are expected? Okay, so BCL1 is going to recognize TGA, TCA, right? So we're gonna scan through the fragment here and see if we can find something like that. Okay, so there it is, TGA, TCA. That's the only one. I don't think there is another TGA, TCA. Okay, so BCL1 will cut it right here. Okay. So what result do you expect? The larger fragment from A, as in like part A, will be cut once, right? To produce 2 smaller fragments, right? Okay. So here is the difficult part. We are going to try to see what happens if you run this on a gel. I think I'm going to be running out of space here. Let me copy it onto another page. Okay. We're going to have 4 lanes. So we're going to draw our gel. 1 well, 2 wells, 3, 4. Oh my goodness. 1, 2, 3, 4. All right. So we have 4 different samples. Lane number 1. Okay, this is lane number 1. Untreated, just the DNA sample alone, right? So if we take this DNA sample, we don't treat it with restriction enzymes, we don't cut it, right? Then it's just going to be one long strand. It's not going to make it very far because it's longest, right? Compared to the treated ones. So that's that. That should be pretty self-explanatory. Lane number 2. DNA sample treated with BAMH. BAMH is the yellow one, right? Okay. So it will cut it into a small one and a long one. Okay, but the long one is not as long as the original, right? So it's probably going to be further down. And then we're going to have a short one at the bottom, right? So that's number 2. Lane number 3, just treated with BCL1. So just the blue cut here. it will also produce a long one and a short one. Okay, but if I compare like this short one with this short one, like this one is a little bit longer, right? Okay, so you would expect it to be a little bit higher up, not as far as the other one. And then this fragment is going to be like somewhere here because it's shorter than the other fragment that was created from the yellow cut. Okay. Now, finally, if you treat it with both BAMH and BCL1, then you will have 3 cuts. Okay. And then you lower it, you will have the same fragment, right? This is the same, right? Same. Because whether you treat it with just BAMH or with both BAMH and BCL1, you will get this piece, right? That piece corresponds to like this one here, right? And it's the same as that one. And then this one that you get is actually this piece that has been chopped into 2. Okay. So when you chop this into 2, you're gonna get this piece, which will correspond to this one. Okay, so these ones are the same. And then you would have a third piece right here. Okay. Does that make sense? It's probably going to take you some time to, work on it yourself and understand it when you do it yourself. Now, fortunately, but you won't have to do this on the test, right? Because it's all multiple choice. But nevertheless, is it's an important exercise for you to go through yourself. Does anyone have any question? Okay. So we're going to wrap up the lesson by talking about gene therapy briefly. The concept of gene therapy involves introducing genetic materials into a person's cell. Okay. And you can introduce genetic material into bodily cells, then we call it somatic gene therapy. You can introduce it into germline cells, like the sperm and the eggs. That would be germline gene therapy. If you do it to the body cells, to the somatic cells, then it will only affect that individuals. They will not be passed on to future generations. But if you do germline cell therapy, then this would affect future generations. And generally, the consensus is we should not be tampering with germline gene therapy because the outcome is out of our control and we are affecting humans that are not yet born, right? And therefore, they never agree to have the their genes are being altered through gene therapy, right? So we don't have their consent, in other words, right, to modify it. So somatic gene therapy is the main area of research. The other one has a lot of ethical concerns. So what is the goal of gene therapy? Okay, basically we have 3 options here. Okay, and we're going to talk about each one of these. Let's start with replacing a non-functional gene. That is the most straightforward one. If you have a cell that has a loss of function mutation, like causing a protein to no longer function, or if you're missing a protein because of a nonsense mutation, right? Then the appropriate thing to do is to put in a functional version of that gene and get the cell to express it, right? Then you're going to be able to have a normally functioning cell, a cell that's going to be functioning normally once again. Now, these type of conditions are usually recessive. Okay? If the condition is dominant, okay, it's harder to fix with gene therapy because you would have 2 copies that you need to fix, right? Okay, like you can't just fix one of the copies. The dominant expressions will always be suppressing the new genes that you're introducing. Okay, so it's typically going to be recessive conditions that we're trying to fix. So that's the first goal, replace non-functional genes. Something to kind of also remember for non-functional, for this particular goal, it's only successful if the effects of the disease are reversible, okay? So in some cases, things are not reversible. Like the damage is already done. If you do this kind of gene therapy, you can slow further progression of the disease, but whatever damage that has been done up to that point in life for that individual, you cannot reverse those things, okay? So things like sickle cell anemia, beta thalassemia, these are hemoglobin-related mutations that cause a person to become anemic. These are good candidates, okay? Sickle cell anemia doesn't really create irreversible damage in the body, right? So once you get the cell to express normal hemoglobin, then the anemia should go away. Okay? Beta thalassemia is more serious because instead of making misfolded hemoglobin, these hemoglobins are like missing a whole chunk in them. Okay, I don't know if you remember, but hemoglobin has 4 chains, right? So in more serious forms of beta thalassemia, like an entire chain is missing due to a nonsense mutation. So those would probably take more of a toll on the body. So if the gene therapy comes in later on in life, right, there could already be some damage to the body. Hemophilia is a bleeding disorder, right? You can't really clot the blood easily. So again, that's a good candidate for gene therapy, right? Once you replace the non-functioning clotting factor, you're going to be able to clot your blood again. Okay, so number 2, second goal, right? Instead of replacing a non-functional gene, you can suppress a gene. Okay, so these are instead for loss of function mutation, these are ideal for gain of function mutation. Okay, so you will introduce what's called like a blocking gene, and the blocking gene that you introduce will suppress the overly active gene, right? An example for this would be blocking an oncogene, right? You remember oncogene? These are genes that are turned on continuously to stimulate a cell cycle, and we don't want that. Okay, we want to suppress those to stop the cancer growth. Sometimes you cannot really reverse the damage. You cannot completely stop the overly expression gene. So you can also try to use gene therapy to alter the course of the clinical course of the condition. Okay, so you can insert what's called like a suicide gene that trigger apoptosis in the target. This would be ideal for cancer treatment. Okay, so if you can just deliver genes, this suicide gene only to cancer cells, and then you turn them on, then all the cancer cells would, die. Right? That would be good. Or you can insert a gene that creates specific markers on the cell, and then the immune system will be trained to destroy them. Okay, so we'll learn more about that in second semester. So here is what it looks like. There's a disease cell. You insert the suicide gene, triggers apoptosis, or it creates some kind of toxin, cause the cell to die. Or you can insert a marker gene that puts special surface antigens on the cell. The immune cells recognize those, and it will destroy the cell. Okay, so those are the 3 goals. Now you can do gene therapy ex vivo or in vivo. Ex vivo means outside of the body, right? So you would extract the cells from the patient's body, such as bone marrow cells. You will modify them in a petri dish, okay? You can insert the genes in the petri dish, and then you reintroduce the cells back into the body. Okay, so that's called ex vivo. This is, you have more control over it, okay? Because you know exactly what you're adding, and you know exactly what kind of cells will be accepting the gene, right? In vivo is doing it in the body. So you would use a vector like a virus, Okay, you put the DNA that you want into the virus, and then you let the virus deliver it, right? This is a little bit unpredictable in some cases. You don't know where the gene is going to get inserted. What if it inserts itself into the middle of another gene? That's going to be problematic, right? What if the virus delivers it to the wrong location, right? So there's like a lot of fine tuning that you need to do in vivo gene therapy. But those are the two options. So like I said, right, there are a couple of challenges here. So target delivery is what I just told you, right? Delivering the gene to the wrong cell, or there could be unforeseen negative health effects because you are disrupting another gene, okay? And sometimes, well, you might have the gene even turning on other genes that are unintended. Okay, like you're not sure because there's so many genes in the body, right? So if you introduce something new, there are these possible off-target side effects. Okay, so here is a vector, a virus. You put a new gene in there, and then the virus is going to deliver it right into the nucleus. That's what's supposed to happen. Now, because we are using a virus as a vector, there could be immune response to some people. Okay, people are reacting to the virus, although nowadays it's very easy to use viruses that are not too immunogenic. Okay, they could strip away all the parts of the virus that causes your immune system to react and then deliver the gene. But still, right, it's a challenge to find a way to deliver genes without over triggering the immune system. Interaction with other genes, I talked about that already, right? It could interfere with genes that are involved in cell division. And if you accidentally turn on a gene that keeps the cell cycle going, that could result in cancer. If you accidentally interrupt a gene that's supposed to stop the cell cycle from progressing, that could also result in cancer. Right? Cost is another thing. Many of these gene therapy are extremely costly, okay, because they are targeting rare genetic conditions, and a lot of times these are tailor approach, because the gene therapy that works for one person is not necessarily going to work for another, since we have unique genetic makeup, right? And so it's going to be very expensive. So like there is this person who actually was blind, and he got his fame from performing on I think it was American's Got Talent. Okay, so he was a blind singer, and then he won, and eventually, right, he got enough fundings from crowdfunding and from supports of the public to do a gene therapy that ultimately cured his blindness. Okay, and it cost $850,000 for the initial treatment. Okay, and then there are additional costs that comes with it afterwards to make sure that it keeps on working, right? So it's not something that can be afforded by everybody. Okay, so like I said, right, so $850,000, and then that's like $425,000 per eye. And then depending on the kind of gene therapy you're trying to do, right, they could run up to millions very easily. Okay, so who pays for it, right? Insurance, are they going to cover it, right? Some pharmaceutical company that specializes in these gene therapy, they say, you don't have to pay for it unless it works. Okay, pay only if it works approach. But what's considered as working? Maybe it works initially, okay, but then the disease regress and it comes back after 2 years, 3 years, right? Do I get my money back, right? Yeah, so that's a little bit tricky. A lot of people turn to crowdfunding for these kind of things. There are numerous examples on the internet, right? So there is this girl who is diagnosed with his name is Mila. Her name is Mila, and she's diagnosed with a rare kind of a nervous system disorder. And the drug costs more than 1.6 million. Right? So like I said, not an average person can afford something like that, right? Okay, so this affordability issue is real. And that is it, right, for the biotechnology unit. Okay, so I did most of the difficult question in the study guide. I'll post the solutions for the rest of it. And next week, right, Monday, you will do the asynchronous. You watch the lecture recording for lecture 10, and then we'll come back and do our Wednesday session where we take up the study guide. And you will also have to do your quiz on Monday. Okay, Monday is not a holiday for the college. Okay, classes are being run as scheduled, so make sure you make note of that. And that's it. Okay, thank you for coming. I'll see you all next Wednesday. Bye for now.